In this module, we will discuss the control of messenger RNA stability. How messenger RNA stability is controlled in the cell, how its time period or its life is controlled, and how it is decided that which RNA has to live. The longer a messenger RNA is present in a cell, you see the messenger RNA is present in the cytoplasm in the cell. And for the longer time if it is present, what happens? The more time it can serve as a template. Messenger RNA, what messenger RNA is? Only a template for a protein synthesis. And if it is present for more time, it means it can serve as a template for synthesis of protein, for synthesis of polypeptides, which are required by the cell from time to time. Now, if a cell is to control the gene expression, it is just as important to regulate the survival of an messenger RNA as it is to regulate the synthesis of that messenger RNA in the first place. So you see, there are two things. One is that survival of the messenger RNA, and the second is the synthesis of the messenger RNA in the first place. That how, if the cell is to control the gene expression, it has to control these two important or regulate these two important factors. One is the synthesis of messenger RNA, and the second one is the degradation or survival of the messenger RNA in the cell. Now, most eukaryotic messenger RNAs, they are relatively long-lived. That is a known fact. Now, long life of a messenger RNA is related to the length of its poly-A tail. The longer the poly-A tail, the longer it will live. Now, when a typical messenger RNA, it leaves the nucleus, it contains a tail of approximately 200 adenosine residues which are present at its tail, that is the messenger RNA which contain this. Now, as an messenger RNA remains in the cytoplasm, its poly-A tail tend to be gradually reduced in length as it is nibbled away by deadenylase enzyme. So what happens? If the messenger RNA, it resides in the cytoplasm for longer time, its poly-A tail, it becomes shortened since it is nibbled away by a deadenylase enzyme which removes its tail. Now, no effect on the stability of messenger RNA is observed until the tail becomes or reduced to approximately 30 residues. If the tail is less than 30 residues, then it becomes inactive, but it should be about 30 residues. Once the tail is shortened, the messenger RNA is degraded rapidly by either of the two pathways. So the tail in the cell, if it becomes less than 30, so survival is difficult and it is destroyed by two pathways which are there. One is the degradation of messenger RNA. It becomes at its 5' end following removal of remaining poly-A tail at the 3' end of the messenger RNA. Now, once the 3' tail is removed, as the 3' end tail is removed, the messenger is decapped and degraded from the 5' end down to the 3' end. So what happens? First 3' end, it is removed. Then from the 5' end, it is degraded to the 3' end. Now deadenylation or decapping and 5' to 3' degradation occur within small transient cytoplasmic granules, which are called bodies. In those granules, this process operates, and that is called bodies. Now, in second pathway, in the alternate messenger RNA degradation pathway, the removal of the poly A tail is followed by continued digestion of messenger RNA from the 3' end. So, in the second, the 3' end, the poly A chain, it is degraded, it is digested from 3' end till to 5' end. In this diagram, the messenger RNA degradation in the mammalian cell has been illustrated. That how there are different mechanisms where poly A, they are degraded. You will see that in the middle, the poly A chain is being nibbled away from 3' end to the 5' end, and eventually it is degraded on the particles which was present in the down, and they further take the poly A to 5' end over there. Now, the digestion of messenger RNAs in the 3' to 5' direction, that is carried out by an exonuclease, that is part of the complex of 3' to 5' exonucleases, which are called exosome. And exosome are, in this diagram you can see at the bottom, those balls are granules which are just at the lower part of the diagram, and these are those enzymes which degrade the poly A chain or that messenger RNA from 3' to 5', and they are the part of enzyme complex and called exosomes. The digestion of messenger RNA in the 3' to 5' direction, that is carried out by an exonuclease. As I said earlier, that the exosomes, they are the part of exonucleases, and this complex 3' to 5' exonucleases, these are called exosomes, remember, and that is very important, that this is degradation occurs on exosomes, and exosome degrade them from 3' to 5'. They keep on degrading into those.